Carbohydrates are essential energy sources for almost every cell on the face of the earth, so it's essential to understand then the biochemistry/metabolism of these important compounds. This lecture will focus on probably the most important carbohydrate, and that's glucose. Glucose is broken down in a pathway called glycolysis, which will be the subject of the first part of this talk, and then I'll finish it with the end product of glycolysis, which is pyruvate, and how it's metabolized further and how that impacts fermentation. Now, in glycolysis, we have the breakdown of glucose, and glycolysis literally means the breakdown of glucose. Glyco referring to the glucose part, and lysis referring to the breakdown. Glycolysis is a primary energy source for cells, but it's not within the pathway itself overly energetic. And as we will see, what glycolysis is important for is providing its end products for further oxidation. Glycolysis is what we call a central metabolic pathway. Now, central metabolic pathway means that it feeds into so many other pathways, and pulling this pathway out of a cell would mean the cell would really have to rearrange everything that it does. The reactions of glycolysis occur in the cytoplasm of eukaryotic cells. The glycolysis occurs in what we call two phases. A first phase in which ATP energy must actually be put into molecules to start the process. The ATP will be recovered later in the second part of glycolysis, where additional ATPs are made, reduced electron carriers are made, and two pyruvates are also made. Pyruvates are a connecting point for other pathways, and pyruvates, the oxidation of pyruvates generates a tremendous amount of energy. Now, glucose structure is seen on the right here. In the body, it's stored, at least in animals, it's stored in the form of glycogen. So the breakdown of glycogen releases essentially glucose for cells to use. In plants, amylose and amylopectin are used as the storage form, but the end product in each case of breakdown is the same. We get glucose that can be used for energy. The advantage of having glucose as a primary energy source for cells, particularly for a multicellular organism, is that it is very readily released from storage, and it's also very easily to travel in the blood. Traveling in the blood is important because fat actually stores more energy per carbon than glucose does. But in a liquid or an aqueous organism such as we are, that fat has to be packaged up in order to travel, and that takes time. Now, another advantage of glucose is that it can be made from very simple precursors, as we will see in another one of these lectures, in a pathway known as gluconeogenesis. Okay, so what I'm going to do now is take you through step by step the individual reactions of glycolysis, discuss a little bit about the enzymes, and then some additional considerations for each reaction as appropriate. In the first reaction, we see glucose being converted into glucose 6-phosphate. This is a very simple reaction catalyzed by an enzyme called hexokinase. You can see that this is one of the reactions that requires an input of ATP in order to jumpstart the process. And the reason that this happens is that the phosphorylation of glucose by itself is not energetically favorable. So the reaction is coupled to an energetically favorable reaction, which is the hydrolysis of ATP to make ADP, and of course the other product being glucose 6-phosphate. This reaction involves the carbon number 6 of glucose, as you can see here, that becomes attached to the phosphate, which is carbon number 6, shown on the glucose 6-phosphate. This is a regulated step of glycolysis. Now, glycolysis has a very unusual system of regulation, and there are some reasons why that's the case. But glycolysis, as we will see, has 3 points of regulation in it, and this first step is one of those points of regulation. Hexokinase, the enzyme, is actually inhibited by its product. So as its product accumulates in cells, hexokinase will quit running this reaction. Now that's important because if a cell is, for example, making glucose, what will happen is glucose 6-phosphate concentration will increase. And if glucose 6-phosphate concentration is increasing, then there's no reason to continue making more of it. Hexokinase gets shut off in the process. The delta G0 prime for this reaction is very favorable because of the hydrolysis of ATP being about minus 16.7 kilojoules per mole, and it goes forward readily. Now, hexokinase is an interesting enzyme. It's found in virtually all cells of the body. It's not specific to glucose, and that's why it's called hexokinase instead of glucokinase. The hexo referring to 6, and the 6 that it's referring to are 6 carbon sugars. So, for example, hexokinase could also work on a 6 carbon sugar like galactose and make galactose 6-phosphate. Glucokinase, which is a related enzyme, has a higher Km, which means it requires a greater amount of glucose to work on, and glucokinase is found in the liver. Now, that turns out to be important because glucose is made in the liver. And so with glucokinase being in the liver and having a higher Km, it means that glucokinase will not be active typically under the glucose concentrations where the liver is making glucose. So that balance is really important. As I said, the hexokinase is inhibited by its product. And one other thing about hexokinase that's very interesting. Hexokinase has a mechanism of action that involves what I like to describe as jaws. And these jaws look sort of like this before the enzyme has bound to its substrate. The two substrates that hexokinase binds to are ATP and glucose. And in this reaction, what happens is once the enzyme has bound the ATP in my top hand and the glucose in my bottom hand, we see the Koechlin-induced fit come into play because the enzyme goes through a clamping down of the jaws, like I'm showing here, which brings the ATP and the glucose into very close physical proximity. This facilitates the movement of the phosphate from ATP to glucose. At that point, the jaws open and the ADP and the glucose 6-phosphate are released. In the second reaction of glycolysis, this simple reaction is catalyzed by an enzyme called phosphoglucoisomerase, and as its name suggests, it's catalyzing an isomerization. The isomerization is simply a rearrangement of the glucose 6-phosphate into a different sugar called fructose 6-phosphate. And though they look very different, fructose and glucose are fairly closely related to each other. This is a readily reversible reaction. It can go forwards and it can go backwards, and the direction that it proceeds will be a function of the concentration of these. Now, reversible reactions are things to think about because when we go to make glucose in the process of gluconeogenesis, some of the reactions are run in the reverse direction. This is one of those reactions. Now, fructose 6-phosphate is converted into fructose 1,6-bisphosphate in the next reaction, and this is probably the most interesting reaction of glycolysis. In this reaction, we're seeing fructose 6-phosphate gaining an additional phosphate and becoming the 1,6-bisphosphate on the site. Well, why is that interesting? It's interesting because of the enzyme that catalyzes that reaction. Phosphofructokinase, also known as PFK1, catalyzes the reaction very much like what hexokinase did. It uses the energy of ATP to put a phosphate onto one of the hydroxyls on fructose 6-phosphate. In this case, the phosphate is going on to hydroxyl on position number 1. Now, this, as we can see, is going through the same mechanism or the same steps that we're going through in the hexokinase reaction. So the phosphate ends up on here. Now, this enzyme is interesting and important because it's a regulatory enzyme. And when I describe regulatory enzymes, what I'm talking about are enzymes that can be turned on or turned off by various things. In the case of hexokinase, for example, it was a regulatory enzyme because it was turned off by its product, which was glucose 6-phosphate. Now, this enzyme is turned off or on by several products, and these products are interesting for us to consider. The delta G0 prime for this reaction is minus 14 kilojoules per mole. It's very favorable in the forward direction, and the reason it's favorable in the forward direction is for the same reason the hexokinase reaction was favorable, and that is that it's coupled to the hydrolysis of ATP. Now, discussing phosphofructokinase and its regulation gives us some interesting perspectives. First of all, there are two materials or two compounds that favor the activation of phosphofructokinase. One of these is AMP. Now, AMP is related to ADP and ATP, the ATP being the triphosphate, the ADP being the diphosphate, and the AMP being the monophosphate. Well, as we talk about the nucleotides, the more phosphates they have, the more energy they have. And AMP is a form of adenosine nucleotide that has very low energy. Cells usually don't have much AMP in them. And the only time they have AMP in them is when they're running out of energy. So when they're running out of energy, they really need to have a boost in that energy. And one of the boosts in that energy is to activate a process that will make energy, such as the breakdown of sugars. So a low energy indicator like AMP encourages the process of glycolysis to occur so that the cell can generate some energy. Now, fructose 2,6-bisphosphate doesn't fit into that scheme. Fructose 2,6-bisphosphate regulation is a little bit different and will be covered in another one of these lectures. ATP inhibits phosphofructokinase. Now, that makes sense because ATP in abundance indicates the cell has got plenty of energy. You don't want to be breaking your sugar down if you have more than enough energy, just like you don't want to be lighting your fireplace in the middle of summer. It doesn't make sense to waste energy that way. Now, how does this happen? If we think about PFK, it has a substrate for ATP. And one of the things that we learn about enzymes is that the greater the substrate concentration, the faster the reaction goes. So how is it that one of the substrates of PFK is inhibiting the enzyme? Well, it turns out that the enzyme actually has two different binding sites for ATP. One of the binding sites is the binding site for the allosteric effects, that is, turning the enzyme off. And the other binding site on the enzyme is for the active site where the reaction is catalyzed. Now, you're probably getting ahead of me at this point, but what happens is these two binding sites have different affinities for ATP. That's important because if we have low energy, we want this reaction to go. That means that the active site, which binds the ATP, has a greater affinity for ATP than the allosteric site. And only at high concentrations of ATP will the allosteric site start to bind ATP. That's a remarkable process, and there are very few enzymes that exhibit this property. In the next enzymatic reaction of glycolysis, there's something unusual that happens, and the cell has to use some tricks in order to get past the barrier. What's the barrier? The barrier is shown very clearly on the bottom of this slide. The delta G0' for this reaction, the conversion of fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, the delta G0' for this reaction is positive 23.9 kilojoules per mole. Now we remember that delta G0' does not determine reaction direction, but it can affect reaction direction pretty readily. That means that this reaction doesn't go very far forward if we start with equal concentrations of everything. Well, of course, the cell has to make this reaction go in order to make glycolysis go, so it uses some tricks. I'm going to show you one of the tricks that it uses. In the reaction catalyzed by aldolase that you can see on the top of the screen, as I noted, there's some tricks that the cell has to use to make this reaction go forwards. This energy barrier that you see here is very significant. Now, reminding ourselves that the delta G is equal to the delta G0', which in this case is 23.9, plus RT times the natural log of all these products on the side, we remember that the delta G for a reaction, if it's negative, will go forwards, but if it's positive, it will go backwards. So the cell needs to have this delta G overall become negative. That means, therefore, that that term on the right, that big mouthful of a term, RT times the natural log of all those things in the parentheses, that term has to be more negative than 23.9 if the reaction is to go forwards. Well, cells can't violate the rules of nature. I've been saying that many times, and it's true. So the cell has to play with the rules of nature to make it happen. And it does by a very interesting process. The process is called pushing and pulling. In pushing a reaction, what a cell does is it increases the amount of reactant. Well, the reactant here is fructose 1,6-bisphosphate, and I've drawn this same equation now to indicate an increase in the concentration of fructose 1,6-bisphosphate. When this happens, that ratio becomes smaller. And the smaller that ratio of all those things in the parentheses becomes, the more negative the natural log will be and the more negative that overall term will be. Well, pushing is one trick that cells can use. The other trick that cells use is pulling. And the way that cells pull a reaction is by removing products, meaning that the cell can actually take away products that in this case are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So how does a cell do that? Well, as we will see when I talk later in glycolysis, fructose 1,6-bisphosphate has some very interesting effects on another enzyme in glycolysis. When fructose 1,6-bisphosphate binds to that enzyme, the reactions ahead of this one start occurring. And when they start occurring, all the reactants in front of this one, including glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, start decreasing. We've accomplished pushing and pulling, and by pushing and pulling, the cell actually makes the delta G for this reaction be negative, and the reaction goes forward. This is a reaction that students of biochemistry frequently like a lot. And the reason that they like this reaction is because dihydroxyacetone phosphate is becoming converted into the other product of the previous reaction, glyceraldehyde 3-phosphate. When this happens, we then have two of every molecule that continues down the pathway of glycolysis. This way, students don't have to learn one pathway for the dihydroxyacetone phosphate and a different pathway for the glyceraldehyde 3-phosphate. We simply have two glyceraldehyde 3-phosphates to follow. The enzyme that catalyzes this reaction is called triose phosphate isomerase, and it's an example of what we call a perfect enzyme. Now, in one of the other lectures that I've given, I've described what perfect enzymes are, but perfect enzymes are enzymes that catalyze reactions amazingly efficiently, amazingly rapidly, with amazingly little material. And the reason that this enzyme does this here is because there's an intermediate that's in the process of making the glyceraldehyde 3-phosphate that's extraordinarily unstable. If that intermediate sits around for any period of time, it will fall apart, and this reaction will not occur. So to keep that from happening, this enzyme has evolved to be very, very rapid in its turnover. So this reaction is also another one of these readily reversible reactions. It doesn't matter too much to the cell which way it goes, and the direction that it goes is going to be a function of the concentration of the intermediates. As you might imagine, this reaction can be reversed in the process of gluconeogenesis. So the delta G0' is a little bit positive, but it's not excessive for the cell to overcome. This, as I noted, is a diffusion-limited enzyme, and I'll remind you that a diffusion-limited enzyme is one whose rate of reaction relies only on the rate with which the substrates can diffuse into the active site. In the next reaction, and I want to remind you that from this point forward, everything will be doubled. So we actually have two glyceraldehyde 3-phosphates, and we're going to produce two molecules of 1,3-bisphosphoglycerate. Now this reaction is important because it's the only oxidation that occurs in glycolysis. We think about glycolysis as being a pathway that's very oxidative, but in fact it's not. There's only one oxidation that occurs. We think about glycolysis being oxidative because of the products that later get oxidized in other pathways. This reaction is an interesting one, okay? We see, for example, on the left we have an aldehyde, and on the right we have an ester. That's the indication that we've had an oxidation. The delta G0' for this reaction is a little positive. It's 6.3 kilojoules per mole, but again, the cell doesn't have a big problem with that. The difference here is that if we look at what's happening in this reaction, a phosphate is being added directly onto the acid to make the ester on the right side. When we did a similar reaction for glucose going to glucose 6-phosphate, we had to couple that to ATP. And the same thing happened when we went from fructose 6-phosphate to fructose 1,6-bisphosphate. We had to use the energy of ATP. So why don't we have to use the energy of ATP here? The answer is because we have an oxidation. Remember that oxidative reactions generate energy, and the energy of this oxidation is helping to favor this reaction going forward. The oxidation, of course, generates electrons, so we see NAD+ becoming NADH. The next reaction of the pathway is where we start to create ATPs. In this reaction, 1,3-bisphosphoglycerate is converted into a molecule called 3-phosphoglycerate. And this is a pretty straightforward reaction. You see the phosphate on number 1, position 1 on the left, being removed and transferred to ADP to make ATP by the enzyme phosphoglycerate kinase. Now, this reaction is interesting in the sense that it's what we call a substrate level phosphorylation. A substrate level phosphorylation occurs when a high energy molecule transfers a phosphate onto ADP to make ATP, which is exactly what you've seen here. Now, many people think, mistakenly, that this is the way most ATP is made in the cell, by simply having a high energy molecule to transfer. It does not occur that way, and in fact, most of the ATP in cells is made by a process called oxidative phosphorylation. This is an unusual reaction in the sense that ATP is made directly. We'll see another one of those later in glycolysis. The delta G0' for this reaction is very energetically favorable, meaning minus 18.9 kilojoules per mole. It moves very strongly to the right, and since we're making an ATP, what that tells us is that there's a lot of energy in 1,3-bisphosphoglycerate in order to make this happen so efficiently. In the next reaction, 3-phosphoglycerate is converted into 2-phosphoglycerate by an enzyme known as phosphoglycerate mutase. And on the surface, this looks like a very simple reaction, but there's actually a lot of complexity to this. We're seeing the phosphate move from position 3 to position 2. However, this molecule has an intermediate that is created that is important to consider. And this intermediate is something that has 2 phosphates. It's called 2,3-BPG. Now, one of the important things about 2,3-BPG is it's a molecule that has a very interesting effect on hemoglobin. Hemoglobin is the oxygen-carrying protein in our blood, and hemoglobin can bind to 2,3-BPG. And when it binds to 2,3-BPG, hemoglobin lets go of its oxygen. Now, that's important because where do we want hemoglobin letting go of its oxygen? We want it to be let go at places where a lot of metabolism is going on, because that's where the oxygen will be needed. And when glycolysis is going a lot, one of the things that it's making as a byproduct is 2,3-BPG. This is a good indicator to the body that, hey, here's where I need the oxygen, and hemoglobin is tuned to give it exactly. Now, the delta G0' for this reaction is about 4.4 kilojoules per mole. It readily goes either direction, as we've seen with some of the other reactions. Well, 2,3-BPG is a pretty important molecule, as you have probably guessed, so let's spend a little bit of time understanding how it is that cells make 2,3-BPG. There are actually 2 ways that cells can make this happen. One of these is by using an intermediate in the previous step of glycolysis. Now, that intermediate was 1,3-BPG. And in glycolysis, you saw that it was converted to 3-phosphoglycerate, and that generated a molecule of ATP. But 1,3-BPG can also be converted by an enzyme called bisphosphoglycerate mutase into 2,3-BPG. Now, this is actually the most common way that cells will make 2,3-BPG, and this enzyme is found in erythrocytes. Erythrocytes being blood cells, and this helps blood cells to make 2,3-BPG to get rid of oxygen. So that's very good. 2,3-BPG can be converted back into 3-phosphoglycerate, as you can see here, by the 2,3-bisphosphoglycerate phosphatase. A phosphatase is an enzyme that takes a phosphate off of something. So if that happens, then what the cell has done is it's done a little sidestep around that substrate level phosphorylation in the last step of glycolysis. It doesn't affect our consideration of glycolysis, however. Now, what I've been telling you, though, is that 3-phosphoglycerate can also make 2,3-bisphosphoglycerate, and the way that that happens is through the unusual mechanism of action of phosphoglycerate mutase, the enzyme that catalyzes the conversion into 2-phosphoglycerate. And so let's think about this. In the first reaction, we saw 2 enzymes that had to convert first to 2,3-bisphosphoglycerate and then back to 3-phosphoglycerate. Now we see one enzyme that's making a byproduct that's 2,3-bisphosphoglycerate. That's a little confusing, but let me tell you how the enzyme works, and maybe you'll better understand how this all happens. In the enzymatic mechanism of going from 3-phosphoglycerate to 2-phosphoglycerate, one of the things that happens is the enzyme has the phosphate on position 3, and it wants to get the phosphate to position 2. Now, you might logically think that the enzyme grabs the phosphate and it moves it up 1. But that's not the way that a mutase works. A mutase instead starts with 3-phosphoglycerate with a phosphate on position 3, and it puts a phosphate on position 2. And then when it's done with that, it takes the phosphate away from position 3, and it's left behind with the phosphate on position 2. So you can see that in the intermediate phase, the molecule has 2 phosphates on it, a phosphate on position 3 and a phosphate on position 2. That's how 2,3-bisphosphoglycerate is an intermediate in this pathway. Now, 2,3-bisphosphoglycerate sometimes escapes the enzyme, and that's how it gets out and goes and flags to the hemoglobin. Here's where the oxygen's needed, because of an accident in the process. If that accident doesn't happen, then of course if that accident doesn't happen, then 2-phosphoglycerate is made and there's no 2,3-BPG. So the slower glycolysis is going, the less likely the accident will be to happen. In the next step of glycolysis, 2-phosphoglycerate is converted into phosphoenol pyruvate. And the acronym for this one's pretty easy. It's called PEP. And you'll remember PEP because it has a lot of PEP. PEP is an intermediate that has a tremendous amount of energy, as we shall see. It has a tremendous amount of PEP. Now, the enzyme that catalyzes this reaction is known as enolase. And in this reaction, what's happening is water is being removed from 2-phosphoglycerate. That's what creates the double bond that we see in phosphoenol pyruvate. And it's that double bond that helps give all the energy to this molecule. The delta G0 prime for this reaction is about 1.8, so it pretty much goes backwards and forwards without any significant difficulties. But PEP, as we shall see, is important for the last step of glycolysis. In the last step of glycolysis, I like to call this reaction the Big Bang of glycolysis. And there's a very good reason why. This reaction is another substrate-level phosphorylation. We see that PEP is converted into another 3-carbon molecule called pyruvate. And in the process, the phosphate is transferred from PEP onto ADP to make ATP. That means that PEP has to have a lot of energy to have that happen. We saw the same thing with 1,3-bisphosphoglycerate going to 3-phosphoglycerate. But what's different here, this substrate phosphorylation has what I call the Big Bang. This substrate level phosphorylation has a delta G0 prime of minus 31.7 kilojoules per mole. That's almost enough energy to make a second ATP. That's amazing. Well, if we think about this, what happens to energy that we don't use? What happens when we have all this energy released? Well, one of the byproducts of inefficiency, whether it's an automobile engine or it's a cell, one of the byproducts of inefficiency is heat. And heat is generated the more we run this reaction, the warmer we get. So I want you to think about the last time you went jogging, and when you were out jogging, you got hot. Why did you get hot? Well, you got hot because you had to run a lot of metabolic pathways, and running those metabolic pathways like this reaction generates heat. Okay, now this reaction, because the delta G0 prime is so negative, is essentially irreversible. This means that if the cell is going to start with pyruvate and make glucose, it actually has to do a little dance around this pathway, and we'll see how that happens in the pathway of gluconeogenesis. Now, glycolysis is, as I said, regulated by 3 enzymes, and the third enzyme after hexokinase and PFK is this reaction. Now, it might seem odd that the last step of the reaction is regulated, but there's a very good reason why. This reaction is inhibited by ATP. ATP, we've learned, is an indicator of high energy. Do we want glycolysis continuing if this cell has high energy? The answer is we don't. But once we make PEP, by golly, this thing's going to go forward because it's pretty hard to stop such an energetic reaction. Regulating a reaction that wants to go forward very bad is important, and the inhibition of this enzyme by ATP is a big consideration. Now, another thing that's important here, alanine's an amino acid. Why is that in there? Well, pyruvate is readily converted into alanine. An excess of alanine indicates you probably have too much pyruvate to begin with. Now, this enzyme is the enzyme that I described before that was activated in that push-pull reaction of aldolase. As fructose 1,6-bisphosphate starts to accumulate, it will activate pyruvate kinase. And what happens when you activate pyruvate kinase? Well, by golly, PEP is going to go to pyruvate. Well, then what's going to go to PEP? 2-phosphoglycerate. And what's going to go to 2-phosphoglycerate? 3-phosphoglycerate. Every reaction preceding the pyruvate kinase reaction is going to be pulled by activating this enzyme. And what's activating it? The molecule that's activating it is the molecule that needs to get converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. It's a perfect setup. The pushing and pulling reaction is signaling ahead to say, we need some help here, guys. And the guy that's helping him is pyruvate kinase. The enzyme is also regulated by phosphorylation. You put a phosphate on this enzyme, it can be turned off. And so regulation, as we will see, is very important in considering how gluconeogenesis, or the synthesis of glucose, is important. Okay, so we've seen overall that glucose plus 2 ADPs plus 2 phosphates plus 2 NADs gives an overall product of 2 pyruvates, 2 ATPs, 2 NADHs, plus the other things that you see here. Remember, we put 2 ATPs in to begin with, but we had 2 reactions at the end, each of which generated an ATP, and since they're doubled, we generated 4. 2 in, 4 out, a net of 2. Now, the last thing I want to consider here is the fate of pyruvate. Pyruvate is the end product of this pathway, but we remember that pathways don't occur in isolation. Pyruvate goes on to something else. And so pyruvate can go on to a variety of things, and the things that it goes into depends upon the circumstances and the cell in which it's found. Okay? So let's look a little bit closer to see what happens with this pathway. As I say, it's not an endpoint, and it's important for a variety of things. It's important to make alanine. It's important for energy, and it's an important source of keeping glycolysis going, as we're going to see in just a second. Okay? Now, let's consider the 3 different things that pyruvate can go to that are shown on this screen. Pyruvate in bacterial cells and yeast cells can be converted into a molecule called acetaldehyde. And acetaldehyde can be converted into ethanol. Beer drinkers, of course, like that reaction because that reaction is known as fermentation, and it's the way that we brew. We make beer, we make wine, and so forth with this reaction. And we make this reaction by closing off the vessel in which the reaction is occurring, and the loss of oxygen in that vessel favors the production of ethanol. Now, you probably didn't think about this, but your cells, that is human, also ferment. Now, fermentation in us is different than it is in yeast and bacteria. Otherwise, we wouldn't have to go out and get beer, right? To convert pyruvate into lactate, right, there's a reason we're doing this, as we will see. But our cells, when they run out of oxygen, they make lactate, also known as lactic acid. And that becomes important for the production of NAD, as we shall see. You noticed in the acetaldehyde to ethanol reaction that NAD was also produced. Now, what about aerobic conditions? Under aerobic conditions, acetyl-CoA is sent to the citric acid cycle for oxidation. Oxidation requires oxygen, and so this makes sense. No oxygen, we have alternative pathways. When we have oxygen, we use it as best we can. Now, the difference between oxidation using the citric acid cycle and oxidation using the fermentation pathways is enormous. If we start with glucose and we go through a fermentation pathway, the net products of that reaction are 2 ATPs. If we start with glucose and we go through the citric acid cycle and oxidize it completely, we get 38 ATPs. That's 19 times more energy under oxygen conditions than under oxygen absence. You can see why cells really like to have oxygen, and you can see why our body is set up to deliver oxygen as much as it can. It doesn't always succeed in that, but it does it as much as it can. Okay, well, why is it that fermentation is even necessary? Well, you probably didn't think about it as I was going through describing the lecture, but I noted that glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate in the only oxidation reaction of glycolysis. That reaction is important because any oxidation requires an electron carrier, and the electron carrier used here is NAD+, which becomes NADH. The cell does not have an infinite amount of NAD. If it converts its NAD into NADH, it has to be able to convert its NADH back to NAD. Fine and dandy, okay? How does a cell do that? Usually the cell does that in a process that requires oxygen. It's called the electron transport system. And what does the electron transport system require? Oxygen. So if there's no oxygen, the electron transport system can't function. And what will happen to the concentration of NADH? It will go high, and the concentration of NAD will go low. Glycolysis can't keep running under those conditions. But imagine you're out jogging and your blood supply is not delivering oxygen as rapidly as you need it. Then what's going to happen in that case? When that happens, then glycolysis is going to stop, and glycolysis is the only thing that's providing those muscle cells with energy. You got a problem. Well, fortunately, muscle cells and yeast cells and all these things have ways of regenerating that NAD, and that's fermentation. We can see here that the end product of glycolysis is pyruvate. So pyruvate in a bacterial system is converted into acetaldehyde, as I noted, and then acetaldehyde is converted into ethanol. That's why the yeast are making ethanol. In the ethanol reaction, NAD plus is produced, and guess where it's used? Back in the glycolysis reaction. So by cycling back and forth between those two reactions, NAD is generated, glycolysis is kept going, and the cell stays alive. Now, animal cells don't convert pyruvate into acetaldehyde. Instead, they convert pyruvate into lactate or lactic acid. You can see this reaction here, catalyzed by lactate dehydrogenase. And the same thing is happening here that was happening in yeast. It takes electrons from NADH and transfers them onto pyruvate to make lactate. That regenerates NAD, and that NAD that's been made goes right back to glycolysis and keeps it going. So we ferment for the same reasons that yeast and bacteria do, but we don't have the same end product. In this set of talks, what I hope I've given you is a good overview of the pathway of glycolysis, the energy needs of the cell, and how energy factors into the overall ways in which the reaction occurs and the overall controls that enable the cell to efficiently make this process happen.